Hi, as you remember that in the last classes, I was introducing the systems biology concepts. And here in the last three classes, I have covered the fundamentals of system biology. And you also remember from the last classes that I will be covering the system biology courses from different books. And some of the books which I will be covering, they are mentioned here, but it is not limited. The reason for such way of presentation is to make it easier for better understanding and also the most relevant topics so that you don't need to go from one book to another to another, but instead you will learn the entire system biology. For example, the genomics based system biology or signaling pathways or cancer systems biology or immunosystems biology or something more. For example, systems pharmacology, you will be also familiar with this course. So please don't forget to subscribe our channel. And in case if you have any questions, you can also write us by email or also make the comment on YouTube channel. Now, the chapter which I'm going to cover here is biological modeling. So basically, you need to understand what is modeling and what are the objectives or aims of this modeling. So biological modeling is basically an attempt to describe in a precise way the understanding of elements of the system of your interest, their states, and their interactions with the other elements. So, in another way, you can also say you want to know the relationship between different elements present in one system. For example, if five people are present in one classroom, then you want to know the relationship between those five people and how much they could influence or affect each other. And for example, with the absence of one of the student, what will be the impact in on the other four people or four students? So this is basically the way to understand at element level or systems level. So this is what you need to understand. In the similar way, you can understand the gene regulatory network, protein network, or signaling pathways networks, and many more things. In addition, if you are interested in drug delivery system, then you can also understand the impact of before and after drug treatment. But here I will start from the very basic. So basically, biological modeling creates mathematical or computational approach to understand or test the hypothesis about and to predict the behavior of the complex biological systems from molecules to the entire ecosystem. These models use equations and the simulations to describe the dynamic interactions, integrating experimental data and providing a framework to study the phenomena like disease progression, ecological dynamics, or cellular signaling. The process involves formulating models using biological knowledge and mathematical tools, then analyzing and validating their predictions in terms of experimental studies. So here you can see the photosynthesis process. So as a layman, when you are in class 10, you understand in photosynthesis that light is required. And if you want to summarize the photosynthesis process, then here water plus carbon dioxide gives the glucose as a product and oxygen in the presence of sunlight only. So this is the real way of representation of photosynthesis in just one reaction, chemical reaction. So you also understand that entire biological process takes place in the form of chemical reaction. So the same thing here, it has been represented with the molecular formula like H2O plus CO2, then as reactants, then the products are formed are C6H12O6, that is glucose, and O2, that is oxygen. So the right side figure, what you are seeing here, photosynthesis in the presence of light for the purpose to get the energy. And then cellular respiration takes place in the absence of light. And this say the other thing is also shown. Now, of course, there is a role of other parts of the plants also, for example, root, for example, leaf, and but I'm not going to discuss those things here because here I'm going to introduce the reactions. But when you will be more experienced systems biologists, then of course you can also include the color greenish, how much green, how deep the green leaf are, and if leaf is only green or the other parts of the trees, for example, the bark of the tree stem is also green, then what, how much energy will be formed and what will be the rate of photosynthesis. So these are the additional things, but as a layman, as a beginner, you need to understand water plus carbon dioxide gives you glucose and oxygen in the presence of light. Or in simplified way, you can say in daylight, during daytime. So now it comes, what are the aims of modeling? So in general, the main aim of mathematical modeling or biological modeling are to gain a deeper understanding of biological system, to test the hypothesis, and finally to predict the behavior which you could expect from that particular biological system to facilitate the design of interventions in medicine and biological advancement or biotechnology. By creating mathematical or computational representations, the researchers can simulate the entire complex processes, identify the key factors, and explore the consequences of changes that would be difficult or impossible to study experimentally. This is the right side figure is the representation of all these things what I discussed in these two slides. Now, here you can think in another way, design a, for example, you need to understand the mechanism of action, generate and test new hypotheses, design new experiments, return to the lab to validate new hypotheses. But when? For this purpose, you need to integrate the new findings into the current model and evaluate them against it, compile and organize the entire results, generate new data from experiments, and continue repeating this process until your goal is achieved for that particular project or purpose. Now, if we want to understand the real aims or specific aims in terms of text, for example, understanding biological systems, this is the primary goal in system biology, or mathematical modeling in biological systems, hypothesis testing, prediction, designing interventions, integrating and quantifying the data, bridging the scales and the gaps, reducing experimentation, discovering general principles. One more thing which has not been mentioned here is also to minimize the time required. So also, when I'm saying reducing experimentation, that means we are saving more and more time and money. Now, for why these modelings are necessary, what type of modeling will be required and when. So, for example, stochastic modeling, I'm saying that why stochastic modeling is required and in which condition. So for this purpose, you need to understand what is stochastic modeling. This I have also discussed in data science course, which I have already uploaded all the videos. You can also see my data science playlist or playgroup. So forecasting the probability of various outcomes under different scenarios or conditions, allowing for random variations in one or more variables. So it's very simple. It is to forecast the probability of various outcomes under different conditions. That means there might be certain factors which could influence, allowing for random variations. So this is here the one of the equation which I'm showing for the linear birth and death process. If we want to predict the population of certain countries, for example, India, Brazil, China, or USA or Europe, then stochastic modeling will one of the finest modeling for this purpose. And the equation which will be used to predict this is here dx by dt equals lambda minus mu of x and x of t. That is here. So lambda and mu are the factors which make it. If you are predicting for next 100 years, then this will be one of the best way. But if you want to calculate those things by hand, or let's say with some manual way, it may take longer time. But in case of mathematical modeling on computer, you can do it in few seconds. So if you want, for example, to add some more and more factors which will affect the birth and death process, as you might have seen in 2019, that is COVID-19, then how much population were decreased? And what was the effect on the population growth? So these are the additional factors. Such factors you can also include. Now, the examples of stochastic plots are mentioned here. One of the figure here is showing one is deterministic plot, another is a stochastic plot. So here you can see how it looks like, and then here this is you can easily understand the deterministic plot and the stochastic plots. So first figure on the left side is showing the 5 plots for the deterministic solution. And with different values of lambda minus mu. And here on the x-axis, it is time point, and y-axis, it is the number, for example, the variable x. In the left side bottom figure, there are another 5 plots with different values of lambda and mu and the variable x. For 5 realizations of a stochastic linear birth and death process, together with the continuous deterministic solution. And here there is continuous solution, deterministic solution. Now this is the another 4 plot which represents different lambda and mu values and different types of those 5 plots. Then you can also understand which would be, what are the factors which could affect more and more. Now, there is another simplest example of implementation which have been published in Nature Communication Biology journal. And this title itself reveals the real application, that is, stochastic modeling reveals the mechanisms of metabolic heterogeneity. So, this stochastic modeling has been applied here to understand the diversity of metabolic processes at different level. And these figures are itself showing the details from the beginning, model, designing the model, that is Michaelis-Menten kinetics, and some plots, then with different mathematical equations, different types of NLD plots, and finally the analysis part. So there are, in the similar way, different applications of different modeling approaches. For example, Boolean systems will be applied for another purpose. Ordinary differential equations will be effective for other purposes. So and the purpose depends on the time and accuracy. So our goal is to get more and more accurate outcome and in less time. So this is what we expect. And for this purpose, we choose different types of model. Now, as I mentioned, in biological system, everything takes place in the form of reaction, chemical reaction. So now you need to understand the chemical reactions. For example, there are a number of ways one could represent a model of biological system. And biologists have traditionally favored diagrammatic schemes coupled with verbal explanations in order to convey quantitative information regarding the mechanisms. At the other extreme, applied mathematicians traditionally prefer to work with systems of ordinary or partial differential equation, that is ODEs and PDEs. So here, there is a general chemical reactions. For example, M1, R1, M2, R2, MR, RR, and then the products. These are reactants. Before arrow, they are reactants, and after arrow, they are products. Where R is the number of reactant, P is the number of products, and capital RI represents the ith reactant molecule. Capital PJ is the jth product molecule, and MI is the number of molecules. So basically, what I want to say here, you need to understand what are the letters representing in the reactions. First thing. Second thing, when you are writing them, so this is the equation in the form of which will be helping you basically to write the codes also, computer code also, and it will help you to also understand the chemical reaction. But you need to start from the basics. For example, here 2P are the product. So, and then it gives the product as P2. So it what does it mean? It means that two molecules of P react together to produce a single molecule of P2, where P has a stoichiometry of 2 and P2 has a stoichiometry of 1. So this is which also you need to understand what are the stoichiometries. So stoichiometries of one are not usually written explicitly, and that is why it is written as P2, not 1P2. Similarly, the reactions for the, so basically this is the forward reaction. But when you are saying it is reversible reaction, then you need to understand the backward or dissociation of this dimer, which is formed here. So P2 now becomes the reactant, which forms two molecules of P. Then you need to now understand again the another way of representation of reversible reaction. And these are quite clear from the basic class 10 or 12 level. So if one of direction predominates over the other, then how are you going to represent? Then in that particular case, you can make one of the arrow as a small to show which one is more dominant. So it is important to remember that the notation for the reversible reaction is simply a convenient shorthand for the two separate reaction processes taking place in the context of discrete stochastic models to be studied. It will not usually be acceptable to replace the two separate reactions by single reaction proceeding at some kind of combined rate. So now you need to understand what is the rate, what is the rate of reaction, what are the kinetics. So this A figure is another example to represent. You can also write in different ways of the signaling reactions or metabolic reactions depending on your choice. Now, it comes to kinetics or rate of reactions. So reaction kinetics is the study of how fast the chemical reaction could take place and what are the factors which could influence. So these are and the things which you need to understand and the variables which can affect the reaction would be temperature, pressure, compositions, and few more factors also. A chemical reaction is usually depicted in the form of chemical equations, as I have shown. And now I'm just going to explain, for example, 2 ADP molecule will form 1 ATP plus 1 AMP. And here, if you this is one reaction, forward reaction, but if you want to say that this is reversible reaction, then you can write in another way with the forward and backward arrows. So now you need to also go for some basic exercise. What does the following reaction notation means? So you better understand all these things. For example, 3A plus 4B are the reactants, and then 3C plus D are the products. This means that during a reaction event, three molecules of A and four molecules of B react to form three molecules of C and one molecule of D. So we need to understand. And then it comes, you also can determine the stoichiometric amount, rate of change, and the stoichiometric coefficient and reaction rates. Now, what is a stoichiometric amount? The stoichiometric amount is defined as the number of molecules of a particular reactant or product taking part in a reaction. The stoichiometric amounts will always be positive numbers, and this you have understood really well. Now, In brief, you can say the stoichiometric amount is the number of molecules of a particular reactant or product taking part in a reaction. That's it. Now, what is reaction kinetics? So, if I just say in one simple sentence, then it is concentration versus time. That is rate of the reaction kinetics. That's it. But if you want to define, it is the study of rates of chemical reaction. So, a study of rates of chemical reaction or how fast the reactants could be converted to the product, or you can also say how fast the products are formed. Then factors, what are the factors which in general are known to influence the reactions or formation of product or the, let's say, conversion of reactants to the products. First one, concentration of the reactants, physical state, surface area of the reactants, temperature at which the reaction is taking place. Presence of a catalyst, and what is catalyst? Catalyst is the substance that increases the rate of a reaction without being utilized or consumed in the overall reaction. So, how will you represent the rate of reaction? Change in the amount of reactants or the product divided by time. This is how. Then there are some simpler general example with some way of representation of rate of reactions. In case of like when you are representing the reactants, then it will be written with a symbol, an additional symbol that is with minus, because reactants are converted to products. So, for example, minus dA upon dt minus dB upon dt, because these are the reactants which will be converted to products or which will be decreasing. And the products are always with the positive sign because they are formed. Now it comes rate of reaction for the formation of ammonia. You can easily, how can you write it? Then here you can see dN2 by dt, then dH2 by dt, but here minus 1 by 3, you can see, because here there is 3 molecules of hydrogen. And then what is formed? 2NH3. 2 molecules of ammonia is formed. So 1 by 2 into dNH3 upon dt. So this is how you can write the chemical reaction in the same way. So you need to remember, for the reactants, you will be using minus symbol also. And for the product, there will be no minus symbol, only positive because it is produced. So you also need to understand the number of molecules will be written how, like, for example, if there are 2 molecules, then we write 1 by 2. If there are 3 molecules, we write 1 by 3. So this is what you need to understand. Then it comes to the slope. What will be the slope? So you have to decide, you have to calculate it, the particular slope at particular time point. That's it. So the slope is here. How you write is D of C, that is concentration divided by time. ET, a specific time point. That's it. And another thing you need to understand, the line or plots, when you are plotting for reactants and the products, the slopes for the products and the reactants must be opposite to each other, which you are seeing here. If the slope of reactant is going down, then at the same rate, the product slope must be going up. That's the only fundamental concept. If it is not going in this way in your computational simulation, that means the way in which you have written the codes for mathematical equation will be definitely wrong. So you are not following the mass action kinetics, because the when you sum it up, all the concentrations of reactants and products at all the time points, it must be equal always to 10, or the amount of initial concentration of the reactant. If the concentration of initial concentration is 10, it must be always 10. If it is 100, it must be always 100 for all the reactants and products. So the amount of products and reactants must be equal to the initial amount of reactants. That's it. Now, what is rate of change? It's very simple. You don't need to go for so many things, so many definitions. The simple equation you need to remember, if rate equals delta S upon delta T, that means the rate of change in concentration or amount of a designated species could be represented as it is here. So delta represents the change. So reactants rates change as reactants are consumed and products are made. The rate of change is better defined as instantaneous change in concentration, or a derivative will be rate equal to ds upon dt. That's it. Then it comes to stoichiometric coefficients, which deals with the static information about the amounts of substances involved in this chemical transformation, whereas kinetics relates to the rates of change that occur in these amounts. To paraphrase a statement, stoichiometry provides the framework within which chemical change takes place irrespective of the forces that bring them about, and by kinetics, the speed of chemical change. So this is what you need to understand. Now, here you can see the rate of change of product. Or you can also call it as progress curve for a simple irreversible reaction. Now, the stoichiometric coefficient refers to the relative amount of substance that is consumed and or produced by a reaction. That's it. Now, the when you are basically looking for such basic terms, first thing, while you are writing or working on a specific project, you need to define the basic or fundamental points related to all the reactions in your project. And then you need to proceed for mathematical modeling or design. So now it comes, if we want to write these stoichiometric calculations for a realistic example, for example, the formation of water from hydrogen and oxygen molecule, then you need to understand in a clear view how you can write them. If you want to write the molecules, then here you can write it. Mass here, amount like this, and then mass in gram, you can write it. So the coefficients in balanced equation gives the ratio of moles of reactants and the product. So this is very simple. So you need to be very clear what is stoichiometry. Then you just go for simple chemical reaction, any simple chemical reaction, and then think about it. What are the number of molecules? What is the number of moles? What are the number of atoms? And what are the reactants? What are the products? You will understand most of the things related to the chemical kinetics or mathematical modeling of a biological system. So now it comes to reaction yields, what you get in hand as product. So you can easily understand, but still you can read what are the reaction yields. What I'm going to say, what are the intensive property and extensive property? Intensive property is a property that does not depend on the quantity of substance. And the example is temperature, density, concentration. But extensive property is a property where it, it depends on the quantity of substance. For example, mass and volume. Now, reaction rates. So, what is reaction rate? You have already seen it. And with which unit it will be represented, it depends on what are the units you are using. Rate of change, you have already heard. So now you need to understand the different way of representation also. But if you are getting confused that there bigger mathematical equations are coming, so don't be afraid, stop it here and then move it to the next part. When you will be needed or required more factors to include, then you can easily adapt it if you are able to develop the small model. So what are the notation? How the concentrations are represented? In many texts, the concentration or molarity of a substance X is denoted by using square brackets, as you have seen. To avoid unnecessary clutter, you need to use one uniform way of annotation. And that is, for example, here the square bracket. Now, elementary rate kinetics. What are the orders of reaction? For example, zero order, first order, second order. For this, you need to understand the elementary reaction. Now, here, the kinetic properties and empirical studies have been shown that the rate of reaction is often proportional to the product of molar concentration of the reactants raised to some power, certain power, 1, 2, 3. For a simple elementary monomolecular reaction, which is here A, reactant gives B reactant. So the rate of reaction V or K is often found to be proportional to the concentration of the species A. So here V equal to K multiplied with A. So what it is here, this property is often called law of mass action. And the corresponding kinetics is called mass action kinetics. And the proportionality constant K is called rate constant. So A is the concentration of reactant and V is the rate of reaction with certain units. Now, recall the rate of change of A is the reaction rate times the stoichiometry coefficient. Since the stoichiometry of A is -1, so the rate of change is given by dA upon dt equals -V or equal to -K into A. So this is very simple. Now, a rate that is proportional to a reactant raised to the first power is called first order with respect to the reactant, but the reactants raised to the zeroth power is called zero order, and the reactants raised to the power of 2 are called second order. So this is what you can see here. And in the similar way, the reaction rates will be different for all the three order reactions. Now, what is half-life? So a common metric that is used to judge the rate of different first order reactions is the half-life. The quantity measures the time taken for half the level of substance to be transformed into product. When half of A has been consumed, we can set A of t in the equation here to 1 by 2 or into A, and then The same thing, half equal to e to the power of raised minus k t half. So these are some of the fundamental terms which you need to know. Now you also can go for more and more derivatives and mathematical equation depending on your need. Now there are some simpler example to represent how you can represent the chemical reaction and the rates of reactions. So they are mentioned here. Now, what is chemical equilibrium? This is what you need to understand. And in the very simple way, you can say the chemical equilibrium when the product formation is reached to saturation phase or when there is no change in the product formation or in the decrease in the reactants concentration. That means we can say the chemical reaction has reached to the equilibrium, that is chemical equilibrium. So here, all the reactants, for example, in a closed system, that is system when it is isolated from the entire surroundings, will tend to go to thermodynamic equilibrium, but in open system it remains different. At equilibrium, the forward and reverse rates of reaction will be equal to, and the net rate will be zero. That's it. And this is what you can say in simplified way when the product formation and reaction reactants decreasing has stopped or has reached to completely unchanged stage, then you can say that it is in the chemical equilibrium stage. Now, what is the equilibrium constant? That is here. The ratio K1 upon K2, if you have two reactions, forward and reverse reaction, then equal to K, that is capital K. This is called equilibrium constant. Here it has been represented with K EQ. So the equilibrium constant is also related to the ratio of the rate of constant K1 upon K2. For general, you can also simplify them according to your need. Now, you can also go for some example how to write the equilibrium constant. Now it comes to dissociation constant. So, for example, we have the bimolecular reaction, that is HA forms H and A. So here it is reversible reaction. Then in this case, the dissociation constant will be Kd equal to H into A divided by HA. So another thing is association constant. It will be in the reverse way. That's it. So the association and dissociation constant will be written in the reverse way. That's it. Reverse means here, for example, in dissociation constant, we write H into A, which are here product. So product divided by the reactant, and here reactant is HA. While in case of association, they are reverse. So we write the HA divided by H into A. That's it. Now, if we want to say what is the mathematical relationship between Kd and Ka, then it is represented by Kd equals 1 upon Ka. That is clear that they are reverse to each other. So you need to understand with different examples also and solve some of the realistic problems, then move to the computational equation for those simplest forms. Now, you need to understand mass action kinetics. I have already defined on the two slides before what is mass action, what is mass action kinetics. So you need to understand them. In closed systems, reaction will tend to equilibrium, whereas reactions occurring in open living cells are generally out of equilibrium. The ratio of the products to the reactants in vivo is called mass action ratio, and that is here. But it is purely or entirely upon you. How do you understand the chemical equations and how do you write them? I will say before you go for law of mass action, you can remind yourself what is law of conservation of energy, where you say there is never ever a loss of energy. In the same way, you need to balance the equations. So the amount of reactants will never be decreased or lost. It will be converted into products. That means you need to think about if the total amount of reactants and the total amounts of products are equal to the initial concentration of all the reactants or not. If yes, then they are perfectly fine. That's it about mass action kinetics. Now, what is are the kinetics across the membranes? For example, from one cell to another cell, compartment A to compartment B, there is always frequent movement of molecules from one cell to another or from cell surface to the cell inside or cytoplasm or nucleus. Then how are you going to define these kinetics? So here you need to think about the system, what are the factors affecting, how you are going to write them up. This is depending on your understanding of the system. Now, how are you going to write the temperature dependencies? I'm not going to explain all these details, which you will be facing or you will be writing while you are developing mathematical model for signaling pathways or for metabolic pathways. Now, how the rate of reaction could look like, how the activation energy, what is activation energy also you need to understand. They are quite simple. So here you can see with this example in the easy way that the energy required here to achieve certain frequency. So for example, here you can see this is threshold level when you cross it up. So here you say this is activation energy needed to achieve the peak of it. And then the energy profile of the reaction. Here the graph shows the potential energy profile. This is potential energy. As the reaction proceeds, the activation energy is the height of the barrier which needs to cross. So that's simple way of definition. Then in the chapter highlight, I will say you need to learn what are the role of potential energy, what is the activation energy, what is net release of energy, lower activation calories, energies in catalyzed reaction, then reaction extent, then what is time, what are the concentrations, when we will say the reaction kinetics, or when we say the dynamics, what is temporal dynamics, what, so these are the terms which you need to understand. What is equilibrium, what is the, for example, stoichiometric coefficients, all these basic things you need to understand. So what I will be doing now, I will go more details and about it. In continuation with this, I will continue further some more things related to this chapter, and that will be a realistic example of enzyme enzymes which you are frequently seeing in signaling and metabolic pathways. Of course, after that, I will also give you the examples of gene activation and the system biology or modeling of the gene activation in terms of computational programming or in terms of mathematical equation. So thank you very much. Please don't forget to subscribe the channel.